My name is Rachel Ritchie. I'm the director of our Agrifood and Natural Resources sector at Genome, BC. And this morning's session is going to carry on from where we left off. Today, we are going to look at some of the exciting applications that are happening in the really sorry, I have to be really close to this, in the agri-food and natural resource sector. It's okay, I got it. I have to stay really close. So we have a great lineup of speakers. So we're going to begin with Stephen Rader, University of Northern BC, Sateesh Kagale from Natural Resource Council, Loren Rieseberg, and then Sally Otto, both from UBC. And following, sorry, hello? Okay. I'll stay away from that mic. This is much better. So we're going to follow the format that the health session took. We'll begin looking at some SynBio applications and look what that's doing in this field. We'll move on to gene editing, and then we're going to add gene drives just for some spice at the end, which will allow us to talk about even more exciting applications in the field. So that's enough for me. Hopefully you heard most of that, and I'll ask Stephen to come up. And then Stephen will give his short presentation, and we'll move through our speakers, and then we'll have a panel session.
Can you hear me? All right, thank you very much, Rachel. So yeah, as Rachel said, we're sort of following on from the earlier session. And I'm just going to give you a little bit of an overview of some of the things that we can do with gene engineering, molecular engineering, and synthetic biology. So as Rachel mentioned, I'm from the University of Northern British Columbia. I'm actually a basic researcher. I study pre-mRNA splicing, but I'm not going to talk about that at all today. The reason I was invited to this session is that we work with a red alga that I will mention during the course of my talk. And we've been developing some tools to allow us to do molecular engineering on this alga, which has some potentially interesting applications. The other connection that I have is I'm the faculty mentor for UNBC's synthetic biology team, and so I'll go into that just briefly in a minute. So I think it's pretty clear from what's been said already, but just to remind you, there's a pretty close parallel between machines and what goes on in cells. So there are machines, and in order to make machines, there are blueprints that have the instructions for making those machines. And there's a two-way relationship between those. The blueprints have the information for making the machine, but then you also need machines to make blueprints. And this is very much the same as what goes on in a cell, where the machines in this case are by and large proteins. And in the nucleus, there's DNA. So the DNA has the instructions for making the proteins, and the proteins are required to make the DNA. So there's the same sort of reciprocal relationship. The other sort of general point that I want to make in helping to think about synthetic biology and what we can do with it is that biology is modular. So one of the kinds of things that cells do all the time is they respond to some external thing which turns on the production of new proteins. And so the different modules that are part of this pathway include the receptor that detects the external signal, the activated gene that starts some new genetic program to respond to that external signal, and then the new protein that's part of that response. So synthetic biology takes the approach that we can mix and match these modules in a way that doesn't happen in biology, even though biology has been going on for at least 2 billion years. There's only a relatively small part of the possible space of combinations that have been tried out in natural systems. And of course, those aren't always the ones that we think would be useful. So just to give you a sense of this, I have a small list of examples of things that the cell might be able to detect. Sugars, amino acids, metals, toxins, explosives, various sorts of things, heat and light, and various responses that the cells could have to respond to that. And so cells, of course, will have, in the natural environment, will have evolved to respond to the things that are useful for their survival, but we can mix and match these things to suit our needs. So for example, we might design cells that move in a certain way in response to the presence of sugar. Or we might design cells that light up in the presence of explosives, or that change color in the presence of heat. One of the interesting things to give you an idea of some of the subtlety and complexity that can be built into this is that you could have a response that actually makes more of the receptor, and so it therefore sensitizes the cell. Or it could make a different kind of receptor, so after one kind of stimulus, it would then become sensitive to a different kind of stimulus. So, as I mentioned, part of my sort of entree into this area is that I've been working with our synthetic biology team, which takes part in this competition that MIT runs every year. It's the International Genetically Engineered Machines Competition, or IGEM. This is just a picture from last year's competition. They had something like 5,000 participants from about 40 different countries. And just to give you a brief flavor of the kinds of things that these student teams do, there was a team that developed an arsenic sensor for drinking water. So they make these small disposable vials that have the engineered bacteria in them, and you can buy them for a few cents. And people who are in places where the drinking water might be contaminated with arsenic simply put the water in this vial and leave it for a couple of hours. And if it stays green, then the water is safe to drink. And if it turns red, then they know that it's contaminated with arsenic and they shouldn't drink it. The team that I worked with at UNBC last year was working on a project aimed at bioremediation. And so the idea was to engineer bacteria so that they would express copper-binding proteins on their surface. And then the idea is that you could work with, for example, mining companies that have to treat wastewater. And it would be hopefully a cheaper and easier way to pull the toxic metals out of the water and collect them someplace where you could dispose of them in a different way. And then in terms of the work that I actually do in my own lab, we've been working with this alga, Cyanidioschizin maroli. And as I mentioned, our interest is really in studying RNA splicing. This turns out to be a very interesting model for that. But we're developing tools that allow us to manipulate the genome. It's an organism that has only been studied by a few labs in the world, and there are not a lot of molecular tools for studying it. And so we're developing ways that we can actually change the genome to allow us to, for example, make bioproducts. You can make biofuels with this. And it grows at very acidic pH. So it's potentially something where we could, again, work with mining companies who have large tailings ponds of very acidic, toxic water full of heavy metals, and either simply use that as a place to grow these algae, or actually use the algae for bioremediation. So that's something that we're sort of looking into for the future. I just want to end up, to finish mentioning that there's this whole sort of parallel universe of kind of do-it-yourself molecular biology going on. And the reason that this is happening is because, as you heard earlier, DNA synthesis has become really inexpensive. So pretty much anybody can order new genes. You can make enzymes at home. The technologies for doing this are pretty straightforward. And there are even these DIY kits. There's one of the IGEM teams from a couple of years ago made a $1,000 kit that includes a thermal cycler and a small centrifuge and basically everything you need to do the molecular biology. And so, we're really not too far from the day where people, in fact, there are already people who are doing this in their kitchen, which of course, raises some interesting questions that hopefully we'll have time to get into in the ethics panel this afternoon. But so just to leave you with this idea that the vision here is to treat biology the way engineers treat electrical circuits and mechanical parts, that we can think about what we want to design and swap the pieces in and out in a way that makes sense. So I hope I've given you a little bit of flavor of what the possibilities are, and I look forward to the questions in the panel session.
Good morning, everyone. First of all, I'd like to thank Catalina and other organizers from Genome BC for inviting me to come and present our work here. I have come from Saskatoon. I work at National Research Council in Saskatoon. And changing the course of the discussion, I'm going to talk about crops a little bit now. So I'm going to talk about wheat, which is one of the widely grown cereal crop species, which provides staple food for almost 35% of the human population. So it's important. It's grown on somewhere between 200 to 250 million acres of land worldwide, and it produces about 730 million tons of product, which is consumed by more than 2.5 billion people. So although these numbers look high, if we talk from the perspective of global food security and how much food we might require in 2050, if we go at current rate of growth, we might be short on food. So to achieve enough food and achieve global food security in 2050, we may have to increase wheat productivity or wheat yields by 60%. So now how can we achieve that? So one possibility is can we increase the acreage of acreage of land on which wheat is grown? But if you look at so I'm giving an example of Western Canada here. If you look at the wheat grown in Western Canada, in the past decade, there is almost 10% decrease in acreage that is under wheat. And then there are many reasons for that. The main reason that comes to mind is low profitability for farmers. So farmers are choosing alternate crops like canola and pulses, which provide them higher profitability. Now, why is profitability low in wheat? And then the reason for that is low productivity. So to achieve food security or improve productivity in wheat, we have to increase wheat productivity by a significant fraction than the 0.9% genetic gain that we're achieving per year now. So if we want to be at this 60% in another 35 years, then we have to increase the genetic gain at least to the point of 2%. So we are at around 0.92 or less than 1%. So we have to almost double the productivity. Now, how can we do that? If we look at how wheat is bred, the conventional breeding, wheat is an inbred crop. So conventional breeding has these different steps. So it involves pre-breeding, where the germplasm is acquired, modified, and adapted to local conditions. And that germplasm is used then to do crossing. And then in the next 5 to 6 generations, the product is selved, and then selections are made for particular traits. So that is breeding part. And then there is finishing part, during which we select for diseases, disease resistance, as well as abiotic stress tolerance, and also we look for quality aspects. So once it passes all those tests, then it goes into registration trial, and then these varieties of the lines that are potentially going to become varieties are they go into registration trial and they are evaluated further. So this entire process takes somewhere between 7 to 15 years. So depending upon how many crops you can get in season. Now this is a very slow and laborious process, and also it depends on random reshuffling of alleles in the lines through genetic recognition. And then this process is limited by genetic variation in the germplasm. So if you do not have genetic variation for a particular trait in the germplasm, then you are not going to achieve it. So some of the complementary or alternative approaches are mutation breeding and hybrid wheat. Mutation breeding has been used in wheat breeding, but again the success rate is very low because again it is random mutagenesis. Whereas hybrid wheat has not been that successful in wheat, and then the reason for that is in wheat, it being an inbred crop, the heterosis for hybrid wheat production is very low. So the gain that we get through heterosis can be achieved through inbreeding, and that's why predominantly wheat is bred through inbred methods. So that's where like CRISPR-Cas technology comes into picture, which offers a subtlety, speed, and high degree of control over the outcome. And then that is because we can precisely modify the genome or genes of our interest and achieve the outcome. So National Research Council realized its potential, and for the past 2-3 years, they have been funding different projects to create a gene editing platform. So I will briefly go through. So we have developed a guide RNA designer for wheat crop, as well as there are different projects that are going on to create genetically edited crop through either transgenic approach or non-transgenic approach, where we are focusing on protoplast-based transformation as well as microspore-based transformation. And also, in this group here, they are focusing on producing gene-edited crops without getting any insertions of the, or getting GMOs. So, my group, like I am more interested in application of CRISPR-Cas technology, and for the past 1 year, we have been working on that. So how can we use CRISPR-Cas technology to reshape wheat breeding? So this is the process I showed for wheat breeding, and during our discussions with wheat breeders, we identified 2 areas where we can apply gene editing. One is the pre-breeding region, where we have the germplasm. So if we have germplasm, for example, what breeders do, they pick lines from germplasm based on the traits of their interest. But sometimes those lines may have some faults. Like for example, you're looking for a disease resistance, but that variety is too tall. So breeders anyway select that line, do the process, and they depend on selections to remove like the tall varieties. But sometimes in the end, like they end up with tall varieties that are not going to be accepted by the producers. So we can use gene editing to improve the germplasm in the beginning itself before it goes into the crossing. And another process is some of the lines that go in registration trials, they not always become varieties. Because in the registration trial, if the quality is not good or any particular trait is not good, then those lines are either removed or they are released as lower class varieties. So those are the difficulties that breeders face. So that is where we can apply CRISPR technology as well. So I will give you, like in the one last slide, I will give you an example how we are using this. And then, so what we are trying to do is, we are trying to improve the germplasm, particularly for the height. And then you already have, so you all know the genes that are responsible for green revolution, the height reducing genes. So these genes can be targeted to produce or improve germplasm for height or reducing the height. And then if you look at the alleles that are responsible for reduced height in wheat, are these RHT alleles. and then they are basically null alleles, which can be easily achieved using CRISPR technology. So this is one of the projects that I'm currently involved in. Another thing is, for example, the registration trial. So there were a couple of varieties in the last year or previous year's trial. They were released, they were bred as premium class CWRS varieties, but they were released as CPS, which is a lower grade class in wheat. And then the only reason for that was the grain hardness or the quality was not that good. So grain hardness is the pin alleles or puro-indolin alleles are the one which confer grain hardness. So for CWRS, we need harder grain, and some of the varieties that were released had softer grains, and that's why they were released as CPS. So can we pick those lines that are released as CPS varieties, and then the breeders would like to release them, or producers even want them to grow as CWRS class because they get higher price for them. So can we take those lines and fix grain hardness in those varieties by mutating or manipulating these alleles, like pin alleles? And then, so the most effective hardness alleles are, one of them is the null allele or hardness locus, and another one is substitution. Substitution is difficult, but right now we are focusing on null allele in the beginning. And if we are successful, then we can look at substitutions. So this is my last slides. And again, as Elizabeth mentioned in the discussion earlier, CRISPR is thought as like very easy method to achieve, but it is not. There are limitations for this technology. And then also, particularly in plant, where like most of the crop species are polyploid, there are many difficulties which affect how we can apply this technology. And then these are some of the limitations that I have listed here. So the efficiency of modification is very low. So when we do gene editing, all of the guide RNAs sometimes work, and sometimes none of the guide RNAs work. So why, we don't know. As well as off-target effects. Particularly in plant, off-target effects are really important, and that is because most of them are polyploid, and then there are many regions where the guide RNAs can go and hit. And also, like, there was a little bit of discussion about the complex architecture or intellectual property landscape, as well as the regulatory hurdles that I'm not going to talk about now, but I think in the afternoon we have a session on that. So with that, I would like to thank my team who are working on this project here, also my collaborators, and then the NRC Gene Editing Working Group, which we work very closely, and then the funding from Canadian Wheat Improvement Program, which is funded by National Research Council. Thank you.
So when Catalina asked me to speak, I initially said no, because we don't do gene editing, although we're planning to, like many people. But she asked me to talk a little more about sort of the prospects for gene editing in plant breeding, particularly industry, the seed industry, taking sunflower as an example. So I think many of you have probably seen this slide, but for those of you who haven't, sunflower is a global oilseed. It's worth about $20 billion annually, the production. It's considered to be a priority food security crop because it produces a healthy oil that's grown widely in developing countries. And it grows well in marginal soils, in extreme environments, and so it's thought to be a crop that will do well with the climates expected in the future. If you look at it, it's obviously, in that lower graph, you can see that it's tiny compared to wheat. I guess that's not showing wheat there, but anyways, for those, oh, it is there. Okay, it's not on my screen. All right, wheat, huge, sunflower, tiny. So you can see the sunflower is about the same range as other important food security crops like cassava and potatoes, but it's nothing like wheat. And as Sateesh says, whether we have enough food in 2050 will depend a lot more on wheat than it will on sunflower. So I thought we should start off with what happened last time. So in the 1990s and early 2000s, sunflower, like most other crops, were fairly major crops, were exposed to genetic engineering, and transgenic cultivars were produced. And you can see in sunflower the usual traits, herbicide, insect and fungal resistance, as well as, because it's an oil crop, seed oil quality. A lot of the usual traits were targeted in sunflower. We worked with companies on a couple of these. One of them was resistance to sclerotinia, which is the worst fungal disease. We also worked some with them on insect resistance, with BT resistance. And there's quite a lot of activity. 9 different companies were involved, as well as 2 government agencies, 1 university. There are a lot more universities involved at the early stages, but the ones that actually went to field trials, I just listed those here. So what happened with all this? Well, nothing. It turns out that not a single GM sunflower was commercialized, despite the literally hundreds of millions of dollars that went into this. And there were 2 reasons why. I'm partly to blame for this, I feel like, because someone else would have done it if I hadn't. In the early 1990s, I published a number of papers showing that sunflowers were highly promiscuous, and the cultivar hybridized widely with wild relatives. And then we also conducted some experiments with a couple of the transgenic cultivars showing that indeed the transgenic gene in one case, in the case of the BT gene, this was work we did with Alison Snow at Ohio State University, it did confer a very large fitness advantage to wild plants. And so would have spread into weedy sunflowers as well as into wild sunflowers. And the concern was that the sunflowers might get more weedy, but also that it might have an impact on insects that only occur on sunflowers. And so that was one thing that came up. But a bigger deal turned out to be money, edible oil marketing. By the time sunflowers were ready to be commercialized, there was increasingly negative perceptions of the technology in Europe. And the sunflower industry, they got together and said, well, we're not going to commercialize because this will give us a competitive advantage in the oil market in Europe. And so this had some consequences. Production plummeted in the Americas over the past sort of 14 years, or actually the past 17 years, and it grew dramatically in Europe. The European market is the bigger one for sunflower, and the gains in Europe actually outweighed the declines in the Americas. So overall it was a good deal, but it meant that for people who do research and breeding on sunflowers in the Americas, it hasn't been a good time. You've either had to move to Europe or get out of the business or sort of try to hang on. And so what's happening is in America, it was sort of like with wheat, sunflowers was pushed aside by transgenic soybean, transgenic canola, and so forth. And of course, when it didn't have to worry about that, it did just fine. So what's going to happen this time? I mean, what's going to happen with edited sunflowers? So the seed companies, as you know, really like the technology. These are some things that Sateesh pointed out, like transgenics, you can actually produce some novel traits, or traits with larger effects than you can get through conventional breeding. Even more important, I think, to the big breeding companies is you can get very strong IP. This will be talked about probably some this afternoon, but natural variants, the IP that you can get on a natural variant is actually quite weak. It's also fast, as Sateesh pointed out, at least in sunflowers, it's thought that it will reduce the breeding cycle by 4 to 5 years. And an important aspect of this is that linkage drag is avoided. And so I've shown this in the down here. What you can see is that often beneficial traits are linked with things that are not adaptive in a cultivated background. And so you have to break those up, and that takes time. Also, of course, it's more precise and predictable than transgenics. Transgenics was a fairly, was not the best technology. And because, in theory, no foreign DNA is required, the hope is that there will be greater public acceptance and a lower regulatory burden. The alternative, of course, is standard crossbreeding. And to get the kinds of productivity advances that we're looking for, you're probably talking about at least some breeding involving wild relatives. This is something that we do, sort of this pre-breeding. And this makes the linkage drag problem even greater because whenever you're breeding with a wild relative, you bring in traits that you don't want and have to be removed afterwards. So what's going to happen? Well, it's going to depend on the public acceptance of edited foods. In the sunflower case, it's going to depend on what happens in Europe. It's also going to depend on how editing is regulated. If the regulatory burden is high, what will happen is the focus will mainly be on a few very high-value traits. This would be the same herbicide resistance, insect resistance, soil quality, and on just major crops. If it's a low regulatory burden, we'll see a lot more creativity, a lot more crops involved, a lot more different traits. I'll give you an example. I sit on the advisory board of a very large French government industry project in France. There are 8 industry partners. And at our advisory board meeting in January, we advised them to actually stick with conventional mutagenesis because there is no regulation. They can commercialize those products without a regulatory burden, whereas with CRISPR-Cas9, there's clearly a good deal of regulatory uncertainty. There's also technological challenges, and Sateesh mentioned this as well. It's more straightforward to achieve CRISPR using sort of conventional transgenic approaches. And what we really want in order to reduce the regulatory burden and to increase public acceptance is to actually achieve DNA-free CRISPR by providing abundant Cas9 protein and guide RNAs directly to cells. Unfortunately, in plants, there's a cell wall. That makes all of this more difficult. I've listed a number of the possible delivery mechanisms here that people are using to achieve DNA-free CRISPR. Protoplast transformation has been used most successfully in sort of a half dozen crops so far, but there are a lot of other different approaches. And so we decided with Sunflower that we would join a collaboration with UC Berkeley's Innovative Genomic Institute. This is the institute headed up by Jennifer Doudna, the discoverer of CRISPR. And we have convinced, I think, a French company, Biogena, to fund the sunflower component of that work. And progress is being made. At Berkeley, they are focusing so far on protoplast transformation, but they also plan to use a gene gun. And at UBC, we had a wonderful undergraduate, Dylan Moxley, last semester, who decided to explore pollen transformations that he's made actually some really excellent progress. And we, for many reasons, we would prefer this approach because it would avoid in vitro regeneration.
I want to switch the topic a little bit and talk about gene drivers. But first, to set the stage, there's been an enormous growth in genetic and genomic data. And this has expanded across the tree of life. So now we have over 3,000 eukaryotic genomes sequenced and over 74,000 prokaryotic genomes. So from this expansion across the tree of life, we're now able to pinpoint genes and discover the genetic elements responsible for such things as climate adaptation in trees. Here, this is from Sally Aiken's work here at UBC on pine and spruce. We're also able, as Loren just mentioned, to improve the breeding and the efficiency of breeding to target the movement of genes bred in from wild crops into the agricultural crops. This expansion of our genomic resources is also leading to new novel discoveries. So, for example, CRISPR, that has been the topic of many talks today, was discovered as effectively an immune system in bacteria. Essentially, little pieces of virus were being stored inside bacterial genomes, and then the Cas9 editing system was able to use these viral segments to target entering viruses, clip them, and basically clip them out. Basically recognize them, and cut them out. So this was an immune system, but then it led to this incredible change in genetic technology, where we can now use the same kind of enzyme, Cas9, the same kind of targeting with a small little chunk of RNA to guide precise cuts anywhere, potentially in any genome. So this is dramatically changing genetic technology by providing a tool that can be more efficient, more precise, and used in a wide variety of organisms. Although it's not always that easy to put into practice, as Loren has told me many times. But now I want to switch to, not only is it allowing new technologies, like changing genetic engineering, but it's also allowing completely different types of possibilities. What I want to talk about is when not only do you have in the cargo for this this Cas9 element that is inserted into the gene, not only are you inserting a new gene of interest or a change of interest, but you're also inserting the very elements, the Cas9 and the targeting RNA needed for the genetic engineering to propagate itself to other locations in the genome. So you insert this element, and you're essentially inserting the genetic engineer as well as the genetic element itself. And so what that means is that, for example, if you had a sperm from a mosquito that carried this element, and it has the cargo includes Cas9 and the guide RNAs, you cross it to a mosquito that doesn't carry that. Well, this guide RNA is then recognizing, oh, look, there's the sequence on this opposite strand, the homolog, cuts that out, and then through homologous DNA repair, you end up copying the cargo and the inserted genetic element and getting 2 gene drives out of it. Okay, so this, let me just walk you through what the implications of that. In normal inheritance, if we have this mosquito with 1 copy of this cassette inherited from the mom and the normal non-edited version inherited from the dad, when it reproduces and produces eggs, 50% of the eggs should carry the maternal copy and 50% the paternal, and that's normal Mendelian segregation. But what happens when gene drive occurs is that during the development of the eggs of this mosquito, that homolog is targeted, cut, and replaced with the gene driver element, causing 100% of the eggs, or some large number of the eggs, to carry this genetically modified element, or genetically altered element. This is an extraordinarily strong selection. From an evolutionary point of view, this going from 50-50 frequency to 100% frequency is incredibly strong. And to give you a sense of how strong that is, I just want to give you a figure that shows over time, over the number of generations, what would happen if with normal Mendelian inheritance, an element started at 5% frequency, would just hover around there. If you inserted this gene and it was disrupting some normal function, you might actually expect it to have a reduced fitness, for example, a 10% fitness loss. But in the case of a gene drive element, we see an entirely different picture. This is instantaneous on an evolutionary timescale, where an allele or gene is spreading through from being nearly absent to nearly complete within 6 generations. That's what I mean by really strong selection. And even if that element, I said, doesn't necessarily get into all of the eggs, even if it's just driven into, say, 75% of the eggs, rather than 100% of the eggs, it's still driven incredibly fast. And a 10% fitness loss barely makes a dent in this spread. So there's huge potential benefits and huge potential risks of gene drive technology like this. First of all, it can be used to kind of undo some of the major detrimental impacts that humans have had on the planet. For example, many bird species in the Hawaiian Islands have gone extinct and are susceptible to extinction because of introduced bird malaria, avian malaria. Invasive species throughout the world are outcompeting many native species. And if we could potentially drive through elements that would either sterilize those invasive species or skew their sex ratio, so say only Y chromosomes and not X chromosomes are inherited, to the point that the whole population is converted into a male-dominated population, leading the population eventually to go extinct, potentially. So we've got the potential to drive in sterilizing elements, the potential to drive in sex ratio distortions, the potential to drive in elements that increase or reduce herbicide sensitivity or pesticide sensitivity. We also have the potential to drive through other elements. For example, to drive through elements that cause mosquitoes to develop faster, and so they can't actually transmit plasmodium, the plasmodium that causes malaria. So the potentials are huge, but and I'm just going. This is just an example of all of these cross-cutting changes that could be effective. We could get rid of avian malaria in Hawaii, for example, or drive through more resistant, more susceptible flies that are carrying diseases and causing food rot. But I think it's probably going to be in these disease vectors that we're going to be most tempted soonest to employ gene drive. So for example, gene drive to eliminate malaria or Zika or dengue or yellow fever. But the risks are huge, and that's what I really want to talk about and start a conversation. Because I think this is something that people aren't talking enough about at various levels, from the public to through scientists and through the government level. So what are the risks? Well, first of all, it could spread beyond the area of interest. You could be targeting an invasive species, but it could spread to the continent in which it's a native species. It could spread beyond the target species. As Loren mentioned, there are hybridization events between species, and this can spread the gene drivers from our target to an unintended species. We could spread different genetic sequences. There could be recombination events that introduce different elements into this driven gene segment. So unintended genes could be driven. A huge unknown is how much resistance will evolve during the spread of these genetic elements. Evolution is an incredibly powerful engine with all the individuals in a population potentially mutating and evolving resistance, but will that be fast or will that be slow relative to the spread of these genetic elements? It's going to depend on what exactly is carried. What is the cargo? For example, if we use in the cargo multiple guide RNAs that target multiple sites, that's going to be very hard to evolve resistance to. So it depends on exactly how it's engineered. And of course, humans have a terrible history of attempting to eliminate pests in ways that have led to many side consequences and unintended consequences. So the risks of gene drive, potentially eliminating mosquitoes or changing their life cycle or changing their demography, and that affecting the bird and bat populations that feed on mosquitoes in ways that we do not have a good enough sense of those interactions and what the side effects will be. And the other thing that I think is, was it Stephen that talked about these do-it-yourself kits? I mean, this is really close to do-it-yourself technology, and that really brings in huge unknowns about this. The first time we see gene drive may not be not be a regulated release. Speaking of regulation, Kevin Esfelt, who's written a lot on this subject, has written that regulation will always be too slow. And that's why I think public discussions need to happen, and we need to be setting our standards through communication before the regulations come in place. And for example, yesterday I double-checked, but Canada.ca does not have any regulations on gene drive at all. Genetically modified elements are already covered, and this would fall under that rubric. But the special issues that need to be considered because of the drive are not currently under regulation. So with that, I will stop. Thank you.